let's move on to the next step. So next we have metaphase. I think this one's the easiest. Metaphase is when the chromosomes will line up on the middle of the cell, and this is called the metaphase plate. I've also seen it called the metaphase plane. But anyway, this is where the chromosomes line up in the middle. And why do you think they line up in the middle? Well, remember, each chromosome here is made of two identical sister chromatid. And they need to go into the new cells. So we need to actually pull them apart. We need to pull them away from each other. So we line them up in the middle of the cell, and then we move on to the next phase, anaphase. And in anaphase, we're pulling apart the sister chromatid. We're separating them. Because these ones here on the top, these are going to go into our new cell over here. And these ones on the bottom, these are going to go into our new cell over here. And plus, even if we could keep this one and this one here, we don't need them because we've already got all of that DNA here. Remember, the DNA is duplicated. So these are copies. We don't need two copies here. We just need one copy over here and one copy over here. Lastly, we have telophase. And during telophase, we start to see the cell visually starting to separate. So this is a really cool picture where you can see telophase beginning. But also during telophase, the main idea with telophase is we're making the new cells now. We're starting to kind of seal them off. And part of that is remaking the nucleus. We need to reform the nuclear envelope. So during telophase, you can start to see the nuclear envelope forming. Because remember, where does the DNA exist in a eukaryotic cell? Well, it lives in the nucleus. So we need to make that nucleus again because we broke the nucleus before. So now we need to make it again. And lastly, we have cytokinesis, which is the actual separation step. It's technically not part of mitosis. It's like the step after mitosis. So cytokinesis is the actual separation. And for animal cells and plant cells, it looks a little bit different. Animal cells are soft, and cytokinesis is pretty easy. We can just kind of pinch these two cells apart. So let's take a look, because I think it's easier with a picture. So this is a picture of two animal cells being split apart. There's two cells. This is towards the end of telophase. And what starts to happen is we need to separate them. So we basically use microfilaments again to form a contracting ring. So these microfilaments are going to squeeze the middle of these two cells here to eventually pull them apart, cut them apart. So this is cytokinesis in animal cells. Okay? But in plant cells, it's different. Remember, plant cells are hard. They have a cell wall. You can't just squeeze them apart. Instead, cytokinesis in a plant cell is totally different. In a plant cell, you have to grow a new cell wall between these two cells. So this is the end of telophase for a plant cell. And you can see what we're doing actually is growing a new cell plate here. And this cell plate is going to become the new cell wall of the new daughter cells. So basically, we just grow a cell wall in between them. While it's growing, it's called a cell plate, but eventually it will grow between them all the way and split apart into two new daughter cells. Very different from what happens in animal cells.